as Sarah said, I'm Jo James. I've been working with APEM for about 2.5 years now, and I'm a principal freshwater consultant. My background is largely in invasive species biology and molecular ecology. Prior to working at APEM, I worked in academia, as well as for the Environment Agency and the National Fishery Services. And I'd just like to pass over to my colleague, James, just to introduce himself to yourselves as well, please.
Yeah, morning all. I'm Dr. James Smith. Been at APEM now for almost 2 years. Similar to Jo, my background is academic previously, but also had a stint in the Environment Agency down in the Riverine side. I will be honest from the forefront in this that I am very much fisheries focused, but do have a generalist overview in eDNA. So any fisheries questions, feel free to post them my way.
Brilliant. Thank you, James. Next slide, please, Sarah.
Sorry, I'm having some technical difficulties. I'm mounting and in the south of England here. Bear with me, everybody. There you go. Sorry.
No problem. Thank you, Sarah. For those of you who aren't familiar with APEM, I just wanted to give you a little bit of an introduction into our background. So APEM actually started off life as a small consultancy back in 1987. As you might be able to tell from our logo, we started off mainly focused on working in the aquatic sector. Since then, APEM has expanded considerably, and we now operate on a global scale, providing advice to government, as well as support on environmental guidelines. And we now also work across the freshwater, marine and terrestrial sectors, and we have our own geospatial division, which provides a range of aerial surveys. Over the last year or so, APEM Group has actually expanded, and we now also include Woodrow, Aquafact, and most recently GoBee. If you'd like to know a bit more about APEM, the services we provide and the sectors we work with, I'd really encourage you to go and visit our website. But for now, let's move on to what we're all here to talk about, and that's eDNA. So next slide, please, Sarah. So what is eDNA? Well, environmental DNA, more commonly referred to as eDNA, refers to the genetic material that can be obtained from an environmental sample. eDNA can be extracted from a range of different types of environmental samples, including water, soil, snow, and sediment. And the DNA within these samples can originate from a range of sources, including waste products like urine, feces, and also mucus, hair, any kind of shed cells, gametes, pollen, and indeed dead and decomposing organisms. So how can we use this in ecology? Next slide, please, Sarah. Thank you. Well, interestingly, the use of eDNA in ecology actually originally centered around the detection of microbial nucleic acids for identifying species of microbes which could not be cultured. Since then, eDNA has been used for exploring microbial diversity in a range of habitats and from a range of environments, including seawater, freshwater, soil, and permafrost. And more recently, the detection of macroorganism eDNA has expanded the potential applications of eDNA in ecology. At the moment, probably the most exploited use of eDNA in ecology is for the detection of specific species, and most commonly these are invasive and non-native species or rare or protected species. And for this particular application, we rely on the use of quantitative PCR or endpoint PCR and sequencing methods. Another fairly commonly used application of eDNA in ecology is in conducting biodiversity assessments, either for general ecological monitoring or for the detection of broader invasive species. So in the case where you may expect multiple closely related species to be present, for example, with North American crayfish species or Dracenid mussels. For this, we rely on the use of meta-barcoding assays. And the third kind of area where eDNA is increasingly also being used is for dietary analysis. So DNA from fecal samples can be used for both the identification of prey items, as well as actually for identifying the host that produced the feces. In this case, we use a combination of qPCR to detect the host, as well as metabarcoding to detect the prey items. Now, it's important to consider that this is not an inclusive list of everything that eDNA can be used for in ecology. We'll talk a bit more about other applications later on in the presentation, but these are the three key areas that I want to focus on today. So you may now be wondering what assays are currently available and how you can use eDNA for your work patterns. So Sarah, next slide, please. Thank you. So the list of commercially available assays is actually quite now substantial and continues to grow. As I mentioned, qPCR methods are typically the most suitable for the detection of specific species or a small number of specific species. And you can see from the lists at the top of the slide here that most or the majority of the currently available commercial assays are either for protected species like your European eel. Great crested newt is a key one, which I'm sure a lot of you who have used eDNA will be familiar with. White claw crayfish, pearl mussel, and also for high priority invasive species, things like signal crayfish and a variety of bivalve species, which are particular concern to the water industry because of biofouling. And there are also qPCR assays available for key pathogens. Again, these are typically focused on those that are high priority in terms of their ability to cause morbidity and mortality to aquatic organisms. Things like Phanomyces astaci, which causes crayfish plague, and also members of the genus Batrachochytrium, which can cause chytridiomycosis in amphibians. So moving away from qPCR, there are also quite a few metabarcoding assays available. These are for conducting broader scale biodiversity assessments, and they're available on a range of different taxonomic levels. So for example, from mammals and amphibians and fish to vertebrates more generally. There are also assays for crayfish and bivalves, as well as macroinvertebrates more generally. And even we can also now look at some bacterial fauna as well. So we've also mentioned it a little bit as well, but for dietary analysis, eDNA is mostly currently used for identifying fecal contents of piscivorous and insectivorous mammals and birds. But it's also been used for host identification. Particularly, this has been used for bat species, so things like Becksteins and brown long-eared bats can actually detect the host from the feces. Okay, so next slide, please. Thank you. So you may be thinking, well, we've already got well-established traditional methods for pretty much, well, for all of these potential applications you've already mentioned. So what are the benefits of us either switching to eDNA or incorporating it into our ways of working? One of the key benefits of eDNA that it's an incredibly sensitive tool. This means it's able to detect species at very low densities. This is particularly beneficial for invasive and non-native species, where early detection is key for successful management, as well as the detection of rare or endangered species, which are often present in small numbers and hence sometimes challenging to detect using traditional methods. Secondly, eDNA is a non-invasive method. So this means that there's less risk of you disturbing these protected native species populations, and also less risk for transfer of invasive and non-native species. Thirdly, sample collection is relatively quick and simple, and this often means that eDNA surveys are less time and resource consuming than their kind of traditional counterparts. As someone who spent hours searching for isolated native crayfish populations and sifting through otter sprains to pick out tiny fish bones, this is something that really resonates with me as something that could be really important. And further to this, because eDNA requires less survey time and less equipment, there is also less potential for attracting potentially unwanted attention at highly visible sites. finally, eDNA does not rely on the taxonomic expertise of the survey. So this often just generally reduces the requirement for surveys to be conducted. So we've talked a little bit so far about the potential applications of eDNA in ecology and the potential benefits of its use. But how reliable is eDNA as a survey method? Well, this is a question we're asked by our clients frequently. It's important to consider that eDNA is still a rapidly evolving field, that its use in ecology is still relatively in its infancy, and there are many unknowns. Indeed, the reliability of eDNA testing will vary between species. Some species are more suited for detection by eDNA than others, related to their propensity to shed eDNA into the environment. Reliability will also vary depending on habitats, and survey design should always be bespoke depending on whether you're working in freshwater or saltwater, in flowing or still water environments. And it also varies depending on the assay type. So as I'm sure you'll be familiar with, different assays will be better or worse established, and some assays will be better tested than others. And relatively recently, a group of authors actually developed a framework for trying to quantify the readiness and reliability of different eDNA assays. So without going into too much detail, I'd just like to give you a quick overview of this framework. Next slide, please. Okay. So Thallinger et al. Recently developed a 5-point scale for assessing assay validation, with levels 1 and 2 covering assay design and optimization, and level 3 being the minimum level of validation required for confidence in positive but critically not negative results. Levels 4 and 5 require some amount of in vitro and in-field testing, as well as for level 5, estimation of detection probabilities by statistical modeling. Now, achieving level 4 or 5 validation does allow for some interpretation of negative results, but this also applies to level 3. Basic assumptions have to be met. So this means things like samples are collected within the correct seasonal survey window for the species of interest, that you test an appropriate number of replicates, and that your negative controls actually return as being negative. Now, providers of commercial eDNA assays should be able to give you an indication of where their assay sits on this scale, but just to enforce it is important to consider the level of validation of the assay you want to use, not only at the design stage, but also at the stage of results in interpretation. As you can see from this, depending on the level will depend on whether or not you can draw any conclusions into any negative results you do receive. So while some assays have now achieved level 3 and above validation, there's an overarching general consensus that more research is needed in this area. Next slide, please, Sarah. So I just want to spend the next bit of time talking to you about a recent research and development project we conducted with Yorkshire Water in collaboration with academic partners at Cardiff University. Now, there are a few elements to this project. Firstly, I wanted to have a look at how reliable eDNA methods are for key species. So this is in terms of the ability for commercially available assays to actually detect species within controlled positive environment. Secondly, we wanted to have a look at how comparable the results of eDNA surveys were to those of traditional monitoring surveys, so things like kick sampling for vertebrates and electrofishing for fish, in terms of species composition and general species richness. And finally, we also had a look into ways in which we could optimize survey design in terms of kind of numbers of samples collected, where these samples should be collected from for different species and for different habitats. But for the focus of this talk, I'd just like to really hone in on the first two aims of the project a little bit. Next slide, please, Sarah. So for the first part of this project, we conducted a series of laboratory trials to test whether or not some commercially available assays for two key invasive species, signal crayfish and killer shrimp, were able to detect these species in controlled positive environment. So we collected signal crayfish and killer shrimp from known wild populations under all appropriate licensing. These animals were then ported to secured facilities within Cardiff University. And after appropriate period of acclimatization, we moved the animals into controlled experimental tanks. We left them for a period of 24 hours, as this is the period which has been shown to be sufficient for eDNA release. And after this, we collected a water sample, which was then sent off for testing by the commercial provider we were using at the time. So what did we find? Well, for signal crayfish, for animals which were held at a density of 1 animal per 10 litre of water, all 3 samples we collected at this density actually returned as positive. So that gives us kind of a good indication that we can have reliable confidence in this method to actually detect signal crayfish. For the killer shrimp assay, whilst we did have some indication of positive results from animals held at a density of 1 animal per litre, we were actually only able to confirm detection in 1 out of the 3 samples we collected. Without going into too much technical detail, this is linked to the fact that, as far as we're aware, the only commercially available assay for killer shrimp that's currently available is an endpoint PCR, which relies on subsequent sequencing for confirmation of detection. Now, because of the product of this PCR and the nature of it, the eDNA company we used had difficulties in generating a confirmatory sequence, and this means that we were only actually able to confirm the positive detection of killer shrimp in 1 of the 3 samples, even though we had evidence or indication of amplification in all 3 of them. So basically from this, we concluded that at the moment, probably the killer shrimp assay we used is not currently suitable for widespread use for killer shrimp monitoring. And this also really just highlights the fact that it's really important to consider the type of assay that's being used, not only at the design stage, but also at the data and results interpretation stage of your project. Okay, so moving on to outside of the laboratory and into the real world, we wanted to have a look at how comparable the results of eDNA assays were to traditional monitoring surveys in terms of species composition and overall richness. And to be able to achieve this, we collected eDNA samples, so water samples for eDNA testing, in parallel with conducting more traditional survey methods. So that's kick sampling for invertebrates and electrofishing for fish. And then we compared the results of these in terms of species composition and overall diversity. And what we found was that the comparability of the results of eDNA and traditional monitoring varied between taxonomic groups. So if we look at this data, which is from the general aquatic invertebrate assessment, what we found was that with eDNA testing, so eDNA bars being the lighter gray on the graphs, we detected an overall greater number of species, but looking over to the graph on my right-hand side, you can see that these were from a fewer number of families. Now, this is likely to be linked to the fact that certain species, so whilst eDNA is actually really sensitive and can offer greater taxonomic resolution for some groups of invertebrates, it's also less likely to detect other groups of invertebrates because they are less likely to shed eDNA into the environment, so things like hard-bodied taxa. It's also key to note here that eDNA will pick up DNA from terrestrial invertebrate species which has run into the aquatic environment. So there is post interpretation of results required if you want to just screen out those species and have a true look at purely aquatic fauna. So from this, we kind of deduced that probably overall at this stage at least, eDNA is more appropriate as a complementary approach to traditional monitoring for general aquatic invertebrate biodiversity assessments. So let's move on to the other organisms we looked at. Next slide, please. So signal crayfish. So whilst these are invertebrates and they are hard-bodied, we did actually get some really promising results from our eDNA testing of signal crayfish. And indeed, we found that at all sites where we knew signal crayfish were present from traditional surveying and observations, we were able to confirm this detection from eDNA testing. We also got some really positive results from our general fish metabarcoding assays, in that from the eDNA results, we were able to pick up all of the species that were detected by traditional monitoring, as well as a couple of extra species. And these often included things like eel and lamprey, which potentially present in lower numbers, which may explain why they were perhaps missed by the traditional surveys. Now, I don't want to steal too much of James's thunder in terms of talking about eDNA and fisheries. So at that point, I'm going to leave it there and pass over to James. Thank you.
Well, thank you, Jo. Just to give everyone a little bit of an overview where I'm going to be coming from this then is, Jo's very much outlined eDNA and I guess the molecular side of things and its use, whereas I'm going to be looking at the actual application that we use within APEM. So the vast majority of our work actually stems from ecological baselines, and we've used this on a range of projects successfully, ranging from large to small. So we're talking large infrastructure projects such as rail and road schemes and water transfer schemes, to much smaller projects such as small habitat improvement works, SSI restoration, and even small-scale construction such as the installation of heat pump exchanges within the lakes. What we've seen internally is that it shows that actually eDNA is a really powerful tool. Similar to how Jo alluded to, it actually can, in some instances, actually be better than conventional methods. And what we have found also is that where you have multiple water bodies, it actually represents a potential cost and normally a significant time-saving element of traditional methods. And it also enables you to survey certain environments which would be normally suboptimal using traditional methods. So if you can go to the next slide, please. So for example, this is, from the outset, this is completely diagrammatic. This isn't actually representative of any scheme, but this is just to show you potential application in kind of in large infrastructure projects. So assume the dashed line would be a proposed rail and new road, and then you've got the actual buffer around it, which could be the impacted area or the associated infrastructure to go with said road or rail. As you can see here, and you might be familiar with this, is actually Kingsbury Water Park, which is just down the road from me, ironically. There are multiple lakes and ponds in here. You're talking in excess of 50 when you count them individually. If you were going to do this with conventional netting or electric fishing where deemed suitable, this would take a team, essentially a day per lake or pond, essentially, maybe even more on some of the larger ones. And even on the larger ones, conventional methods themselves just simply aren't adequate enough to inform the species assemblage. So this is where eDNA really comes into its own, and it can really provide a good overview of the species assemblage in very quick time. Next slide, please. And likewise, on a small scale, again, this is actually just a local lake I used to fish when I was a child. But I always remember that the fish used to hang very much in that back of the macrophyte bed, not for the pond. And it's just, the reason I show this is that if you're going to use conventional seine netting techniques, for example, to do a stock assessment here, I know from local knowledge that fish are going to be in the north, and you're not going to get a net through that. So actually, you're likely to very much underestimate the population size and potentially the species. Now, again, this is where eDNA comes into its own, and we've used this a lot with councils, looking to try and do habitat improvement works, mostly linked to things like eutrophication, and trying to do desilting to try and bring back ecological benefits of the pond in the park, say. And you can see here that you can quite easily get a small inflatable boat around the margins to do that northern region, or even taking it from the bankside. And by doing this, you should be able to pull a holistic assessment of the actual fish species or any other of the species that you're looking at, depending on the assay you want to use. Next slide, please. So from an ecological baseline perspective, the results that we get, you can, as Jo has alluded to previously, you can do it as a targeted on an individual basis. Now, this is normally done for protected or invasive species, and this utilizes qPCR. And normally what we use in schemes like this, normally in the construction industry, is looking at the whole species assemblage. And this is to a certain degree is to identify protected species, but given that the range of, let's say, fish in this instance, that's my background, it's so varied, you can have over 10 or more species in a pond, you may have multiple protected species, so it's more cost effective to do the method barcoding route in this instance. And what we've seen is that these results can actually be used to be informed ecological baselines and where approved in a regulatory setting. So, for example, Great Crested New eDNA is now a standard method, which now runs alongside other methods such as torching and fossil trapping. Next slide, please. So, from a regulator point of view, it has kind of been a mixed bag in terms of the history. So this isn't due to regulators being slow with the uptake, it's more a cautious approach, let's say. This is primarily due, as again, Joe alluded to, that eDNA is still very much in its infancy, and although it's at a very good commercial stage at the moment, the actual amount of viable assays that are available to provide suitable results is still relatively limited. However, steps have been taken to try and implement this into routine biological monitoring programs. And I know a lot of the people on the call are from the Environment Agency, so you'll probably be aware of this. For those that aren't, dietary DNA was previously used to do WFD settings, class settings that is, but was later pulled due to inaccuracies, and that's gone back to the manual microscope method. GCN, as I alluded to before, that the eDNA methods are now well established and are used commonly throughout schemes like this that I mentioned before, especially on large schemes where you've got multiple ponds. But more recently, what we're seeing a trend towards is actually now fish eDNA. So although that has had a long-standing, widely accepted assay which provides very good results, I guess at the moment people don't really know how to utilise it, but it seems that we're kind of moving in that right direction. So can we go to the next slide, please? So what I wanted to share with you is a couple of examples that we've seen very recently that we will be using for certain clients. Now, I know a lot of you will be familiar with this, but I just thought this is in relation to the Water Framework Directive, which I spoke about previously. So there's just a very brief overview that, for those that don't know, in general is to try and make every water body to a good ecological status with the next cycle ending in 2027. Now, the reason I bring up this in particular is that it is important because, if you go to the next slide, please. Back in September last year, actually, the fish element from lakes has just been accepted to use eDNA as the accepted method now. So its kind of driver behind this was that it was shown that eDNA simply outperforms traditional methods, and that's when you think about it, it's pretty obvious, really, isn't it? You've got nets which may have lengths of up to 4 or 5 meters, let's say, in depth, and then if you're sampling lakes which are deeper than that, then you're never going to get a true representation of the fish there, particularly if you're going to get some of the char and corallid species which are prominently benefiting deep lakes. It's never going to be a true representation of what that lake is actually doing. So the way that this is done is that you calculate by comparing the observed species against the expected occurrence, and the expected occurrence is based on baseline productivity of the lake, which is calculated using mean depth and mean alkalinity. And from that, it will give you a number, and it will tell you on how productive that is. And based on that scale, it will give you a predicted species assemblage within said lake. Now, these species are grouped into different groupings here. So you've got brown trout on its own, you've got perch and zander grouped into one, roach on its own, salmon, char, and corals, and carp and bream. Again, this is all linked to productivity, so a lot of you will be aware carp and bream are synonymous with high productivity lakes, eutrophication and such. So in this instance, if you were to get a large number of carp and bream, then this would indicate that you've probably got a poor lake, and vice versa. If you were to have salmon, char, and corals in a lake, then that would be shown to have good quality. Next slide, please. So this is actually quite a complex system, methodology, sorry, and actually can be quite expensive. So although previously I may have said that it can be more cost beneficial, this may, from a WFD perspective, may need to be offset or do a very good cost benefit analysis to see what is most suitable in this instance. The reason why this is that you require 20 transects around the lake, which is equidistant equidistantly set around the perimeter, and each of these transepts will be one individual kit and will be one individual laboratory analysis cost associated with it. The other thing you've got to take into consideration is that within each of these you need to take 5 subsamples, and that is pulled together into the one main sample, which will then be filtered and sent off to analysis. Now, the way that it's calculated is that it's done on a proportional basis on how many of your samples contain those indicator species in the groupings. So if you were to find to have, as I said before, most of your samples contain carbon-brine, then it will be deemed a lower status than it would be if you were to have salmon, chlorids, or char in there. Next slide, please. So the second an interesting involvement that I guess we have with eDNA now is in relation to the ill regulations. Again, I won't teach you the subjects because I know a lot of people on here with the regulators, and you'll probably have much more of an understanding than I do. But in brief, it's just in response to the collapse of European eel recruitment within, well, globally, and from that they've created eel management plans which looks to boost stocks by doing numerous things, such as making alterations to structures in the barrier, such as installing fish passage or complete removal, screening intakes and outfalls to prevent fish entrainment, stopping European eels, and also monitoring programs to see if these types of schemes are actually working. Next slide, please. So where this comes into eDNA, to realise that that wasn't said in the first slide, is that we've recently been working a lot with the Environment Agency and water companies in relation to their reservoir draw-offs. Now, they will need to have a level of fish screening deemed suitable to the fish assemblage within it. And what we've been tasked to do is use eDNA to actually prove presence absence of European eel within these water bodies, and that in itself will then inform their legal obligations under the eel regulations. So, for example, if they don't have screening, they will need screening. If their screening isn't suitable, then it might need to be improved to meet best practice. So, for example, we might need 2mm mesh screens, for instance. It's really important to stress here that it's not a golden bullet, and as Joe alluded to, there's several factors you need to take into consideration, such as the actual survey plan itself, the ecology of the species that you're actually trying to target. So, for example, when we do these types of surveys, we actually use an amended version of the WFD lake assessment that I previously just spoke about, but we will actually target eel-specific habitats. And instead of just focusing solely on shallow water, we will look at spatial variability in both the reservoir and in terms of depth, just to make sure that we have enough spatial coverage to adequately assess presence and absence. Okay, and I think I'll be passing back over to you now, Joe.
Thank you, James. Brilliant. So just to finish off our talk, I just wanted to give you a bit of a flavour for the other ways in which eDNA is either currently being used in ecology or is in development for. One area where we have already used eDNA is for microbial source tracking, and broadly this refers to a suite of methods which are used to detect the source of fecal contamination in environmental samples, and this is something we have recently used for our bathing water assessments. Secondly, another way in which eDNA may be used is for carrying out population abundance assessments. However, studies conducted to date which have tried to look at whether or not there is a correlation between the amount of eDNA detected and the number of species or number of animals of a species within a population present have yielded mixed results. And I think the general consensus is at the moment that eDNA technology is not quite ready for this application, but watch the space. Thirdly, as well as providing insight on existing populations, eDNA testing of carbon-dated deep sediment samples could be used to help inform reconstruction of ancient ecosystems. Now, from an invasive species perspective, this could be interesting because it could be used to help pinpoint the time at which that particular species was introduced into the ecosystem. Fourthly, meta-barcoding can be used for investigating plant-pollinator interactions and looking at which plant species are most commonly visited by pollinators. And this is potentially particularly important in context of the fact that many pollinator species are in global decline. Lastly, many microbial communities respond to pollution, and therefore assessment of microbial communities can be used as an indicator of pollution. So whilst pollutants themselves don't really have DNA, we can use eDNA as a way of monitoring microbial communities, and hence potentially getting an indication of any pollution going on. Next slide, please. So there, I just wanted to give you a bit more of a few more ideas of potential ways in which you might be able to incorporate eDNA into your ways of working. And I'd just like to finish by with a few take-home messages. So hopefully between myself and James, we've managed to demonstrate that eDNA has great potential, particularly for species detection, particularly for invasive and protected species, and for carrying out biodiversity assessments, particularly for those taxonomic groups which are particularly suited for detection by eDNA, like fish. Because of the numerous factors that can influence the results of eDNA testing, those sample collection and survey design should always be bespoke, should be species dependent, should be habitat dependent, and should be ideally formulated in consultation with experts in the field. And because of this, or linked to this, having a molecular understanding is useful, not only at the assay design or the survey design stage, but also at the results interpretation stage. As we've discussed, eDNA is still a developing tool, and its use does require caveating. In most cases, we currently consider it as something which should be used as a very useful complement to traditional monitoring, rather than as a replacement, but obviously James mentioned a few where there may be potential exceptions to this. Overall, we see that the demand for future use of eDNA is likely to continually increase, and we hope that by delivering this webinar, we've at least in some way given everyone a bit more confidence in and some ideas for using eDNA as part of their projects going forward. Next slide, please. So I guess all that's left for me to do is to say thank you to everyone for listening, and I'll pass back over to Sarah to chair any questions. Thank you.
Thanks, Jo. Thanks, James. Before we go on to questions, and we do already have a couple in the chat, we're going to launch another poll. So based on what you've now learned today from Jo and James, would you consider commissioning an eDNA survey? So Tom, if you could launch that survey, please. Terrific. That's brilliant. Great, thanks, guys. So, as I mentioned, there's a couple of questions in the chat, Jo and James. So I'll read them out. I'm not sure which one of you is going to be best placed to answer. So sending out there for both of you. Edward Davies has said, could eDNA analysis be used as part of a biology assessment following a pollution event to show differences above and below a point source in a riverine environment?
Good question. Happy to have a stab at that, James, and then maybe pass over to you if you've got anything to add. The thing about eDNA in flowing waters is that eDNA is obviously mobile, and so anything, you will get movement of eDNA from upstream to downstream, and this might kind of influence your ability to potentially be able to look at community differences on such a small scale. So I guess that it depends on maybe how far you have to assess how far upstream you might need to go to get kind of a representative community, and how far downstream you need to go to get a community results which is not influenced by the upstream. But then I guess it all depends. It have to be bespoke, I think, depending on kind of how variable the habitat and how likely the community is to change in response to habitat variation, as well as in relation to pollution. So I think there's potentials, but you would need to probably factor in quite a few different caveats in terms of movement of eDNA and kind of use local knowledge of the ecosystem that you're expecting in that water course as well. Anything to add on that, James?
Just one thing to say that would be important from a pollution stance is actually the impact of the pollution itself. So if it's going to be a very large scale pollution impact, then the effect's going to be more than something small scale. And where I'm getting with this is that the impact downstream could be greater or less, and the impact from which, let's say, it dilutes out and it goes back to pre-pollution conditions is very, very important, because you may see a recolonization of species, not the sessile species such as macroinvertebrates, but more the mobile species such as fish may recolonize that area once the biological and abiotic conditions are suitable. So if you were going to try and plan on doing some pollution event, you'd have to probably be there as it's happening to get a real sense of it, I would think. I would think it would be difficult to try and, given the amount of variables, I think it would be difficult to try and use that data with a very high degree of confidence. I think you would probably still use traditional methods primarily in relation to pollution events.
Brilliant, thanks both. Oh, sorry, Joe.
Oh, I was just going to add, potentially what it might be useful for is long-term kind of recovery monitoring. I know that eDNA sometimes is used for, well, James talked about lots of construction projects it's used for, but also looking at kind of community recovery. So it would be very simple in terms of it would just tell you what species are present at a given time point. But if you did that over repeated time points, you could potentially use it to try and get an insight into species recovery. Yeah, so just a thought.
Great, thanks both. We've also had a request from Sarah. Would we be able to get a link reference for the eDNA validation framework? Sarah, yes, not a problem. We'll add the link in the email that will come out next week along with the video, so you can click straight through. Next question is from Joe. Question for the speakers. Is APEM interested in or developing eDNA methodologies for pathogen surveillance within important freshwater fish stocks?
Yeah, I'll take this one, Joe. I guess it's probably important to stress that our position is we don't actually develop eDNA assays in-house. That's done with cooperating partners. So normally, if an assay wants to be developed, that's normally led by demand. So if there is a demand for pathogen surveillance, then yes, that's fine. I think we've already seen, we've already got pathogen assays out there for other species. And again, it's very much dependent on demand from the consumer. But we would be more than happy. It would be no different in terms of the methodology, essentially, than the other eDNA methods. So it is viable. It's just dependent on those laboratories actually creating those assays for us to use.
Great, thank you, James. Another question. Do you think we will get to the point of mitigating false positives and be able to sample rivers more effectively for different species? Or should we be thinking about the biome level and move away from species?
So sorry, is that a question on whether or not we'll ever get to a stage where we can rule out false positives? I think that's my interpretation of that. I mean, in terms of false positives and false negatives, really that comes back to that validation scale, and it really depends on how well validated your assay is. And to achieve level 5 validation, which is what ideally all assays should be aiming for, you need to go through quite a lot of rigorous testing in field and in the lab and in silico. So kind of simulations with statistics. And it also needs to be tested for cross-reference against other species. So if it's a specific assay, you'll need to test whether or not you get amplification of closely related species. So I think it may be possible, but given a number of factors that influence eDNA results, in terms of including things like, if you accidentally disturb a sediment when you're sampling a water course sediment, eDNA can be locked in sediment for several hundreds or thousands of years. So there's always that chance that, you're going to resuspend something and detect something which is no longer actually there. So I think it would be tricky to rule it out entirely. Don't know if you've got anything to add on that, James.
No, I think you're absolutely right in relation to false positives. It's going to be very difficult, I think, to actually 100% remove them. And I think that's where that expert analysis needs to come into it, to understand if there is a contamination at the site, if there's contamination at the lab, who have processed the results or not. But one thing I did want to pick up is on when you said about sampling rivers more effectively, what we're currently seeing is actually a kind of a move somewhat into automated samplers. We've tried a couple of these with varying successes at the moment, but again, it's very much at the infancy. But what that will give you is, if you can deploy them in the river, you can leave them in for a number of hours and you'll get eDNA records over a much longer spatial and temporal extent. So that's very dependent upon the river itself, and the dynamics, eDNA degradation, how much eDNA upstream will get to your sampler and such. But it should provide potentially better results than the normal pump sampler that we use, the manual filtration ones, that is, sorry. So there is definitely movements towards making eDNA more suitable in rivers. I just think we're very much in the early stages with it at the moment, but I think in a few years' time, I think we'll probably be somewhere there. So I think we're also relatively close in terms of getting biomass estimates as well. Again, there's a lot of variables involved in that, much of the sampling process, but it does seem it's moving at a very fast pace. So it wouldn't surprise me if in the next 5 years or so, we're probably at the preliminary stages again of trying to put out biomass estimates as well.
Brilliant, thanks, James. Another question from Edward. When planning an eDNA analysis, how would someone go about assessing the reliability and accuracy of the assays analyses that are offered? You mentioned that the confidence for the killer shrimp assessment wasn't that high, but presumably is still available commercially. I think that's one for you.
I think that's a very good question. My first suggestion would be obviously to see if an eDNA company can give you an indication of the level of validation of the method that or the assay that they're using, and provide you with as much information as possible on what that means. But it also does require some kind of, I guess this is where the molecular understanding comes into it, and kind of anything where you're reliant on an endpoint PCR assay. So it's important to get an understanding of, firstly, what assay you need, and secondly, what assay the company intends to use for, because for the single species detection, there are two very viable options that you can go down, either the qPCR, which tends to be specific, just detects one species and you get your result at the end of the qPCR. Whereas you can also have your more conventional qPCR methods where you get your PCR product, which you then need to send for sequencing to work out what species you've got present. So essentially your sequence will be compared to a centralized repository of sequences to come back with a match. So ask for the information, ask them what assay they're using. Anything where you've got sequencing involved, that's where obviously there will be an additional step and you won't get your results straight away. So it's important to consider, if you are using an endpoint PCR, there is that additional step. And anything where your PCR product is kind of small, that's where you start to get more difficulties in terms of amplifying or sequencing the PCR product. So it's a bit about kind of understanding of the molecular methods being used, understanding of how the labs in question have tested those, whether or not they've tested them in the field, if they've tested them for cross-reaction against other species, how much they've used them, as much information as you can ask for from those companies. And I think ideally you probably need some consultation from somebody with some molecular insight, just to help you out with kind of at least being aware of the potential pitfalls with different, or limitations of different assay types. So I think it's a moving feast, as James said. Things are tending to be moving very rapidly, but really we just need a few more kind of research and development type projects like the one we did with Yorkshire Water to have a look at how these things actually can work in a commercial setting, or on a regulatory monitoring basis where they need to be used frequently and the results need to be relatively fast and reliable.
Brilliant, thanks, Jo. Next question is from Yasmin. When you are trying to find the presence of a species in the field where water movement is high and eDNA pollution can vary, Would sampling water in certain seasons, i.e. during zebra mussel spawning or crayfish molts, be preferable, or are the assays sensitive for this not to matter?
Another good question.
We're testing you today, Jo.
From my knowledge. So for species where seasonal sampling is required, your eDNA provider should always give you a recommended window for surveying. So I think it tends to be that things like crayfish, obviously they are less active over winter and they tend to kind of go into, say, a semi-hibernation. So the window of surveys usually runs from kind of April to October. So when they're more active and you're more likely to get that a higher amount of eDNA release. Very interestingly, I'll talk about crayfish because that's my background, but there have been some studies which have suggested that you are actually only able to detect crayfish in the field during the time when they're carrying eggs. So I'm sure some of you have been familiar with how crayfish carry eggs, but they carry them basically under their abdomen, and that means they're constantly grooming and cleaning them and constantly releasing kind of mucus and greater amounts of DNA into the environment. So there have been some suggestions that crayfish detection at kind of low population densities perhaps only possible during those periods where the females are carrying eggs. We did actually have a bit of a look into that in part of the Yorkshire Water Project, and we didn't actually find much any difference in detectability between females carrying eggs and not carrying eggs. So it's something which needs more of a kind of more research is probably the answer. But as you say, molting will also be a good point of them releasing more eDNA into the environment. But you also get massive spikes of eDNA when animals die, so obviously that's something that's happening continually. So I think general response is to always stick with the eDNA assay providers recommended seasonal windows. You always need to consider your target species in terms of whether or not it's something which is what I tend to call leaky, so whether or not it has a high propensity of to shed DNA into the environment, in terms of, you know, considerations for that in terms of picking it up with the eDNA. But at this stage, there's some research into seasonality, but for most species, in terms of, you know, key life history traits, we're basically just going on predicted trends rather than necessarily quantified information. So stick with the guidance and watch this space in terms of the research, I think, is my suggestion at the moment.
Brilliant.
Sorry, just to add, sorry, Sarah, on that. I think it's a very good question, and it raises a lot of interesting points. And it's one thing that may be able to come over that, having that high dilution impact out of suboptimal seasons later might be the automated samples, for example. But when you're sampling greater volumes of water over a longer period of time, you've just got a higher propensity to detect that eDNA in much smaller quantities. But I just also wanted to flag, just to everybody in the room, that again, a lot of this very much depends on ecological knowledge and life histories of the target species or population, let's say. Because, for example, with fish populations, if the kind of reverse is true, where during spawning there's such a gamete release that actually you can mask the other species in smaller and lower abundances, just simply because the metabarcode analysis isn't as sensitive as the qPCR. So again, it's understanding both the ecology and kind of the molecular analysis side of it to really bespokely tailor something to your own needs. I think that was just worth flagging at this instance as a kind of side note.